Dr. D, Dr. D, Dr. D. Dr. D, Dr. D, Dr. D. Dr. D, Dr. D, Dr. D. Explain stuff.
Hey everyone, Dr. D here, and in this video we are going to be doing our exam review for exam 1 for Biology 2421 with the Brock book here. Let's go ahead and get started. Chapter 1, the microbial world. We want to know obviously what is microbiology. Let's see, let's hop right into it. One thing I would like you to know is some feature of all cells. You should know that all cells possess a plasma membrane. All cells possess a cytoplasm, ribosomes. All cells possess DNA. Remember, prokaryotes possess one circularized chromosome in the form of a nucleoid, whereas eukaryotes possess membrane-bound nucleus with multiple chromosomes inside. So all organisms have at least one chromosome of DNA. Prokaryotes have the nucleoid, eukaryotes have nucleus. Remember that eukaryotes have membrane-bound organelles. Prokaryotes do not. All right. Please understand some of these properties of all cells, that all cells undergo metabolism, growth, evolution. Now, one thing that's really interesting that I pointed out in class that you might want to know for the exam is that according to our studies in genomics, archaea and eukarya have a more common ancestor than either does to bacteria. So, archaea and eukarya are more closely related genetically, are more closely related than either is to bacteria. You may want to know that. You should know the three domains of life: bacteria, archaea, eukarya. Let's see. What I'm doing, by the way, I'm flipping through and I'm highlighting things that are essential to know for the exam. I may miss some things, obviously please study all the material for the exam. Don't just go off of this review only. What I'm doing in this review is just at least highlighting some of the things that you should really know. But again, it's not a totally comprehensive review. I don't have time to explain all of the material over again. But again, my main point in this review is just to get you an idea of some of the concepts to master. But the best way to score the best on the exam that you can is to study all the material. All right. Maybe you should know that we have more microorganisms living on our surfaces then we have our own number of cells. So this includes the surfaces of our skin, the surface of our mouth, the surface of our esophagus, the stomach, small intestine, large intestine. Remember, these are still our surfaces. But we should not have any microorganisms within our tissues. Recall that. That was a big point we made. You should know, if you see a name, please know the name and know their contribution. For example, Robert Hooke published Micrographia. He published some of the first microorganisms. Leeuwenhoek was known for his pinhole microscope, where he was able to describe some of the early prokaryotes. Note the difference between magnification and resolution. Recall, magnification is the ability to make an object larger, whereas resolution is the ability to distinguish between two adjacent spots, two adjacent objects. Then we talked about light microscopy. Please know the difference between bright field, phase contrast, dark field, and fluorescence microscopy, and DIC microscopy. Let's go ahead and talk about that. How do you get total magnification? Remember, total magnification is the magnification of the ocular times the magnification of the objective lens, which, remember, the objective lens can be changed. It's on a rotating nose piece. bright field microscopy, remember with bright field microscopy that you don't get a lot of contrast, but you do get color. You can see color, and it's best to stain for better contrast. You should know what contrast means. It's the ability to distinguish a specimen against the background. You should know what is a basic dye. Remember, basic dyes are positively charged, and they stick to negatively charged cell surfaces. So I would want to know that. Know what a differential stain is, and that gram stains are an example of a differential stain. With phase contrast microscopy, perhaps know that this allows you to gain better contrast in unstained living cells using a phase ring. So because you're using this phase ring, it results in an image that stands dark against the light background. Then with dark field microscopy, remember this one, light reaches specimen, and only the light that strikes the specimen is collected in the objective. So you have a dark background with a light specimen, and that's best for looking at motility, unstained samples, looking at living samples. With fluorescence microscopy, I want to know that with fluorescence microscopy, I need to use a fluorophore or a fluorescent molecule to tag my protein or DNA so that I can visualize it. I cannot do fluorescence microscopy without using fluorescent molecules to tag the specimen I want to see. With DIC microscopy, I want to know that I'm using a polarizer, so polarized light, in order to see a three-dimensional image like this. You see these yeast cells, I see a three-dimensional image using a polarizer with polarized light. With confocal scanning laser microscopy, confocal microscopy, I'm able to generate a three-dimensional fluorescent image. So DIC is a three-dimensional image using regular light. Confocal is a three-dimensional computer rendering using fluorescence light, fluorescence microscopy. And then we move on to electron microscopes. You should definitely know the difference between electron microscopes and light microscopes. With electron microscopes, remember, you are not using light, you are using electrons. You are not using glass lenses, you are using electromagnets. The electrons are not flying through air, they are flying through a vacuum, so you must produce a vacuum for it to work better. Remember, there's two forms of electron microscopy. There is TEM, or transmission electron microscope, and SEM, the scanning electron microscope. Let's discuss each. This is your TEM microscope. And what I want you to know about the TEM microscope is that you have to section your specimens. You have to slice them up. And then how do you stain for electron microscopy? You should know you don't use basic dyes for electron microscopy. You use heavy metals such as silver in order to coat your specimen. Okay, so your specimen is coated with gold or silver, some heavy metal, and that's how you stain for TEM. Okay, and SEM, by the way, for electron microscopy. So you can see with TEM, you're sectioning the sample and staining with a heavy metal. And by the way, I want you to know that TEM has better magnification and resolution than SEM. So what's SEM? Remember, in this case, you are coating again with a heavy metal such as gold. But here you do not section your sample, you are looking at topography, which means surface structures. Next, we're looking at some of these early pioneers. You should know the contributions of Louis Pasteur. You should know he's responsible for developing the process of pasteurization. He's responsible for disproving spontaneous generation with his swan neck flask experiment. He is responsible for helping to develop vaccines. He is responsible for understanding how alcoholic fermentation works and the contribution of microorganisms for alcoholic fermentation. So a real powerful person, powerful pioneer in the field of microbiology. Next, we talked about Koch, and you should know Koch's contributions. First of all, a lot of the members of his lab, Walter Hess, his wife Fanny Hess, Richard Petrie, these people contributed immensely to culture conditions, aseptic technique, isolating conditions, media, the Petri dish itself with agar, all of that, those techniques that we use to this day in the lab. But additionally, what you need to know about Koch is his other major contribution, which was his 4 postulates. You should know the 4 postulates of Koch and how he used the postulates. For what reason, you should know the postulates allowed Koch to link a specific organism, a specific species of bacteria, for instance, with a specific disease. So he was able to link a specific organism, for example, Bacillus anthracis, to the disease anthrax poisoning. Okay, we should know Ignaz Semmelweis and his hand washing techniques that he brought to bear. We should know about Lister, Joseph Lister, and his, he was a surgeon who treated his surgical instruments with phenol and had great results there. We should know about Behring and his, he was the first to describe the virus. We should know about Winogradsky, who proposed the concept of chemolithotrophy. Okay, and that leads us to the end of chapter 1. These are all important concepts to know. Again, please do know the Koch's postulates and why they were so useful. And otherwise, it's a really straightforward chapter and just really interesting but straightforward concepts to know for chapter 1, which makes sense. It's just an introductory chapter. So let's take a quick little break time with Gizmo and Wicket, come back with chapter 2, and then we'll finish off with chapter 3 in a little while. Hey everyone, welcome back from break time with Gizmo and Wicket. Let's go ahead and get started with chapter 2 of this exam review. Chapter 2 deals with microbial cell structure and function. So let me hop through this chapter and point out some important points. Morphology. Please know the different morphologies of a prokaryotic cell: coccus shape, bacillus shape, spirillum, spirochete. All of these shapes, budding or appendaged, filamentous. Please know that smaller cells have a larger surface area to volume ratio than larger cells, and this allows for better diffusion through the cell, meaning better absorption of nutrients into the cell, better excretion of wastes out of the cell. So smaller cell size results in better surface to volume ratios. What else we should know about the membrane? And we should know that the phospholipid bilayer in both bacteria and eukarya have an ester linkage between the glycerol head and the 2 fatty acid tails. It's an ester linkage. Why is that important? Well, because archaea, if you remember, have an ether linkage instead. That's probably good to know. And obviously know the structure of a fat, or of a phospholipid, I should say. You've got your glycerol head, 2 fatty acid tails. You also have a phosphate group and another charge group such as ethanolamine or choline, etc. So here you have the phospholipid bilayer. Know that there are many transmembrane proteins inside of a phospholipid bilayer. Remember, archaeal membranes use an ether linkage instead of an ester linkage. Archaeal lipids also have isoprenes, which are like these branched fatty acids. You see the fatty acid tails have branches on them, little methyl groups, which are the isoprene units. You also have the ester bonds instead of the ether. You also have the ether bonds instead of the ester bonds in archaea. So that's good to know. You can even have these monolipid structures, the tetraethers they call them, tetraethers, which forms a mono layer in the archaeal cell membrane. You should definitely know the functions of the cytoplasmic membrane in prokaryotes. So not only is it a permeability barrier, but it is also a protein anchor, and it is used for energy conservation. So you can use it to power things in the cell. What are 3 examples here of energy conservation? There's a proton gradient that forms, and that proton gradient, you can use the proton motive force to make ATP through ATP synthase, to promote active transport, and to drive the flagella. So you can use this proton gradient that's formed to do many things. Next, you should know the difference between a gram-negative cell wall and a gram-positive cell wall. Very important. What is a cell wall made out of? You should know the subunits of the peptidoglycan cell wall. You've got N-acetylglucosamine. Remember, this is abbreviated NAG. And N-acetylmuramic acid, abbreviated NAM. And these are modified glucose that are linked together with the beta-1,4-glycosidic linkage. Then you have the short peptide tail made up the amino acids L-alanine, D-alanine, D-glutamic acid, and either L-lysine or diaminopimelic acid, or DAP. Here's what it looks like. This is the basic subunit of a peptidoglycan cell wall. Again, you have the NAG, NAM disaccharide, followed by the short peptide tail, again of L-alanine, D-glutamic acid, either L-lysine or diaminopimelic acid, D-alanine. So this together is called the glycan tetrapeptide. That's the basic unit. This whole thing is the basic unit of the peptidoglycan cell wall. Remember that in gram-negative bacterium, the tails, these peptide tails, cross-link directly, whereas in gram-positives, the peptide tails cross-link via glycine interbridge. Recall that the peptidoglycan strands, or cables, they're called peptidoglycan cables, form a helical structure, and the backbone of this helix is sugar, the NAG-NAM sugars, and sticking out of each side are the short peptides. So let's discuss the difference between a gram-positive cell and a gram-negative cell. Do you recall a gram-positive cell? You've got your plasma membrane. Outside of that, you have your thick peptidoglycan layer. You also have these lipoteichoic acids as well as regular teichoic acids. And don't forget the reason you have these teichoic acids. Remember, acid means negatively charged because they bind divalent metal ions such as calcium, magnesium, and they also help with increased flexibility of cell wall. Okay, so again, they bind to calcium and magnesium prior to their transport and increase flexibility of the cell wall. That's the function of the teichoic acids. So definitely know the structure of the gram-positive cell wall and know these teichoic acids and the thick peptidoglycan layer. Also remember that there are a few prokaryotes that lack cell walls. These are the mycoplasmas for bacteria and the thermoplasmas for archaea. These are a group of pathogenic bacteria related to gram-positives, these mycoplasmas. And then we talked about gram-negatives. You remember the gram-negatives? So what does a gram-negative cell wall look like? You've got the plasma membrane, but then you have a thin layer of peptidoglycan. And then outside of that, you have another membrane called the outer membrane. And don't forget, the outer part of the outer membrane, you have these structures that are long. You see these hexagonal structures. These are actually sugars. Recall that these are LPS, lipopolysaccharide. These are lipopolysaccharides. The lipid part of the lipopolysaccharide is called lipid A, and I want you to know that lipid A is very toxic to your body, toxic to cells, your human cells. And then you have the core polysaccharide, so a bunch of sugars, and then you have the O-specific polysaccharide, a bunch more sugars. So the outer part of the outer membrane includes LPS. The space between the plasma membrane and the outer membrane is called the periplasm. You also have these porins. You see these triplet circles here. These are called porins. They allow substances to go in and out of the outer cell membrane, porins. All right, please know about capsules and slime layers. These are polysaccharide layers. Remember that capsules are tightly attached to the cell, whereas slime layers are loosely attached to the cell. Please know some of these functions of the capsules and slime layers listed on this slide. Know about the fimbriae and pili. These are filamentous protein structures. You should know that fimbriae are these short fibers that help with attachment to surfaces. You should know that there's different kinds of pili, like the conjugative or sex pili or sex pilus, which you could remember during conjugation between bacterium, the sex pilus forms. But you could also have a type 4 pili for adherence to host tissue and to support twitching motility. Inclusions, you should know that inclusions are storage reservoirs inside of the cell. You should know the process of sporulation for forming endospores. Definitely know the difference, or not the difference, but that Bacillus and Clostridium are two main genuses that form endospores. These are the two main endospore forming genus of bacterium. You should know that the vegetative cell is the growing cell, and the vegetative cell can form an endospore. Let's see. Please know the layers of the endospore: exosporium, spore coat, cortex. You should know about dipicolinic acid and how it conjugates with calcium in order to dehydrate the endospore and to protect DNA. You should also know about the small acid-soluble spore proteins, SASPs, and how they also help bind to the DNA and protect the DNA against insults. So please know the structure of an endospore. Please know the stages of sporulation. They're listed out here, numbers 1 through 7. Step by step, the stages of sporulation. Please know about the various flagellar arrangements: monotrichous, amphitrichous, lopotrichous, peritrichous. This will also help you on the exam, on your practical exam. Okay, flagella. You should know that the filament of flagella is made of flagellin. So here the filament of flagella is made of flagellin. Then you should know that you also have a hook. The hook connects the flagella, the filament, to the basal body. The basal body is the part of the flagella that's embedded in the cell wall. You should also know about gliding motility. Please know about the different forms of taxis. What's the difference between chemotaxis, phototaxis, aerotaxis, osmotaxis, and hydrotaxis? Please know the difference between those. And then with chemotaxis, please know that counterclockwise rotation usually is associated with runs, forward runs, whereas clockwise rotation of the flagella is associated with tumble. Remember, the run and tumble behavior of flagellated bacteria. Counterclockwise run, clockwise rotation, tumble. And when you have an attractant, you make your way towards the attractant through a series of runs and tumbles. All right, that's it for Chapter 2. Let's take another little quick time with Gizmo and Wicket. Come back strong to finish off Chapter 3 of this review and get prepared for Exam 1. Let's do it. Alright everyone, welcome back from break time with Gizmo and Wicket. Let's carry on with Chapter 3, Microbial Metabolism, to finish off this review strong. Remember that if you need any kind of refresher at all about cellular respiration, fermentation, or photosynthesis, these videos will help you to get that refresher. So let's go ahead and get started. Please know what metabolism is. Catabolic reactions release energy, anabolic reactions require energy. Know the difference between the macronutrients and micronutrients. Specifically, let me show you something. You should know that all of your biomolecules, your proteins, your nucleic acids, your carbohydrates, your lipids, all of them require carbon, hydrogen, and oxygen. So you should know that carbon and hydrogen and oxygen are the elements that are used in all of your biomolecules. Additionally, you should know nitrogen, sulfur, and phosphorus are very important as well in these biomolecules. Now, remember these long intimidating names, photolithoautotrophs, etc. I do want you to know these concepts for the exam. So, for example, I do want you to know that heterotrophs obtain their carbon from preformed organic molecules, whereas autotrophs obtain their carbon from the air in CO2. I want you to know that phototrophs use light for energy, whereas chemotrophs obtain their energy from oxidation of a compound, whether it be organic or inorganic. I want you to know that lithotrophs obtain their electrons from inorganic substances, whereas organotrophs obtain their electrons from organic substances. Okay, therefore, if I give you this, for example, if I say on the exam, a chemolithoautotroph, where does it get its carbon from? Or a chemolithoautotroph, where does it get its energy from? You'll be able to do that. Or I could say another way on the exam, I could say, what would you call an organism that gets its carbon from organic sources, its energy from inorganic chemicals, and its electrons from an inorganic electron donor? Then if I give you a list like this, you could pick out that would be a chemolithoheterotroph, for instance. All right, so hopping into cellular respiration. Again, just because I'm flipping through these slides doesn't mean don't know them or don't understand these concepts, but at the same time, I'm trying to hop to those concepts that I really need you to master for the exam for sure. So recall that we have glycolysis is the first step here of cellular respiration. Oh, but before we get there, I really need you to know for the exam, this is a big an important point here, the difference between aerobic respiration, anaerobic respiration, and fermentation. Please know the difference between the three, and please know that none of these are the same. In aerobic respiration, you have glycolysis, oxidation of pyruvate, you have Krebs cycle, and you have oxidative phosphorylation. However, your terminal electron acceptor is oxygen. Does that make sense? So in aerobic respiration, your terminal electron acceptor is oxygen after the electron transport chain. In anaerobic respiration, it's identical, except your terminal electron acceptor is electronegative molecule other than oxygen, but otherwise it's identical. So aerobic cellular respiration is identical to anaerobic respiration. The only difference is the terminal electron acceptor at the end of the electron transport chain. Whereas in fermentation, I want you to know that fermentation is a result of being low on your terminal electron acceptor. If you are an aerobic respirator, if you're running out of oxygen, that's when you switch to fermentation. If you are an anaerobic respirator, if you're low on whatever your terminal electron acceptor is, such as sulfate, nitrate, CO2, fumarate, whatever it is, once you're low on that terminal electron acceptor, even as an anaerobic respirator, you will switch to fermentation. And I want you to know that during fermentation, the only process that is going on is glycolysis. I want you to know that during fermentation, you yield only 2 net ATP. I want you to know that during aerobic respiration, you net 32 net ATP as a prokaryote, 30 net ATP as a eukaryote. I want you to know that during anaerobic respiration, you do not typically yield as many ATP as aerobic respiration. So in anaerobic respiration, it's not unusual to yield only 20-something ATP per glucose molecule. Does that make sense? So you get the most ATP per glucose with aerobic respiration. Next, you get a fewer number, but still a significant number with anaerobic respiration due to the oxidative nature of these molecules. They're not quite as strong as oxygen. And then you only get 2 net ATP with fermentation. I need you to know all of those things. Again, with aerobic respiration, the final electron acceptor at the end of the electron transport chain is oxygen. Anaerobic respiration, the final electron acceptor at the end of the electron transport chain is something other than oxygen. It's nitrate, sulfate, CO2, iron, something else, right? So these are the terminal electron acceptors. And both of these, aerobic respiration and anaerobic respiration, can switch to fermentation so long as you are running out of your terminal electron acceptor. Next, I want you to know about glycolysis. Remember, we covered this. I don't need you to know each and every step of glycolysis. I do want you to know these things. Ready? I want you to know that glycolysis occurs in the cytoplasm. I want you to know that you invest 2 ATP during the investment phase, and then you make 4 ATP during the payoff phase, so you net 2 ATP. I also want you to know that you yield 2 NADH. And I need you to know that your glucose gets broken into 2 molecules of pyruvate during glycolysis. Okay, so that's what I need you to know about glycolysis. You get 2 NADH, 2 net ATP, and 2 molecules of pyruvate. All right, next you have what's called pyruvate oxidation. Remember this step up here where I'm highlighting right here. Your 2 pyruvates go to oxidation of pyruvate. The pyruvate becomes oxidized. That forms NADH. Your 3-carbon pyruvate also releases a CO2 molecule, becoming a 2-carbon molecule. And with the addition of a coenzyme A, your pyruvate is now called acetyl-CoA. It's a 2-carbon molecule called acetyl-CoA. Okay, so did you see that? What I need you to know is that next your pyruvate gets oxidized to acetyl-CoA. This forms 2 NADH, 2 CO2, and 2 acetyl-CoA. Next, I want you to know about the Krebs cycle or TCA cycle or citric acid cycle. They're all the same thing. This is the TCA cycle, citric acid cycle, Krebs cycle. So how does this work? Your incoming acetyl-CoA, and recall you have 2 incoming acetyl-CoA. Your acetyl-CoA gets connected to oxaloacetate. Remember, oxaloacetate is the substance that has the reason the Krebs cycle is a cycle is because what's developed at the end, what's formed at the end is oxaloacetate. And the reason for oxaloacetate is to bind to the incoming acyl-CoA to form this molecule, citrate, also known as citric acid, where the cycle gets its name, citric acid cycle. Next, again, I don't need you to know each and every little step, but I do want you to know that, your citrate is ultimately oxidized, forming 1, 2, 3 NADH. And recall, you have 2 acyl-CoAs, so you get 2 rounds of the Krebs cycle. That means you get 6 NADH, 2 ATP through substrate level phosphorylation, and 2 FADH2 through substrate level phosphorylation. You also have 1, 2 CO2 times 2, 4 CO2 produced as well. Again, what do I want you to know? I want you to know that 2 acyl-CoA enter the Krebs cycle per glucose. I want you to know that 6 NADH are formed. I want you to know that 2 FADH2 are formed. I want you to know that 2 ATP are formed. And I want you to know that 4 CO2 are released. And lastly, I would like you to know that the reason this is a cycle is because at the end of this cycle, you have to remake oxaloacetate. Oxaloacetate must be remade. Why? Because it's going to join up with the next incoming acyl-CoA to start the cycle again. And don't forget, I also I forgot to mention, but the ATP that was formed during glycolysis was formed formed by substrate level phosphorylation. You should know this term, substrate level phosphorylation. What does substrate level phosphorylation mean? It means you're using a high energy intermediate such as this 1,3-bisphosphoglycerate. You're using an organic molecule, a high energy organic molecule, in order to make ATP through phosphorylation of ADP to ATP. So this ATP was made by substrate level phosphorylation. See, this is the energy molecule that made this ATP with substrate level phosphorylation. During the Krebs cycle, this ATP or GTP, it was made by substrate level phosphorylation. All right, let's keep going. All right, hopping through. Okay, this is the electron transport chain of prokaryotes. I want you to know that the electron transport chain of prokaryotes is found in the plasma membrane, and so is the ATP synthase as well. Okay, what else do I want you to know? You should know how ATP synthase functions. Remember, protons force their way through ATP synthase, causing ATP synthase, the top part, to spin, and that provides the energy. That's the exergonic process that provides the energy for the other part of ATP synthase, this part, to produce ATP by joining ADP and inorganic phosphate to form ATP. This process is known as oxidative phosphorylation, not substrate level phosphorylation. And by the way, you should know, where's the proton gradient high in prokaryotes versus eukaryotes? Remember, in eukaryotes, the ATP synthase is in the inner membrane of the mitochondria. Well, the proton concentration is high in the intermembrane space, okay? And the protons force their way into the matrix to make ATP. But in prokaryotes, I want you to know that the proton concentration is high outside of the cell, and those protons force their way through into the cell, driving ATP synthase. Another thing I want you to be aware of is this E sub O. Remember this term, reduction potential. EO, standard reduction potential. A negative value for EO, or standard reduction potential, a negative value means not very electronegative, easily oxidated. A positive value means highly electronegative. These are good electron takers, good oxidizers. So glucose is a very negative value on the, on the reduction potential scale, because it is so easy to oxidize glucose. However, oxygen has a very positive value on this scale because it is great at stealing electrons. It is highly electronegative. So you can see here that in the electron transport chain from complex 1, 2, 3, and 4 down to oxygen, you are going from a very negative value for standard reduction potential, and you're going more and more positive. And that's why the electrons flow through the electron transport chain. So what do I need you to know for the exam? I need you to know that during the process of cellular respiration, electrons flow from molecules of a lower standard reduction potential, a lower EO value, to molecules of higher and higher and higher EO value until they reach oxygen, which has the highest EO value. And that's how the spontaneous flow of electrons occurs. That spontaneous flow of electrons releases energy that's used by pumps 1, 3, and 4 to pump protons actively. That's why you end up with so many protons in the intermembrane space or outside of the cell in prokaryotes. Those protons then force their way through the ATP synthase through chemiosmosis, exerting a proton motive force, spinning ATP synthase, and that's that chemiosmosis is the spontaneous process that releases the energy required to do something that requires work, and that is oxidative phosphorylation, taking ADP and inorganic phosphate to produce ATP. I want you to know on the exam how bacterial and archaeal electron transport chains might differ from those of eukaryotes. They could have different electron carriers, so different electron carriers in the electron transport chain. They could be branched, so the electron transport chain could have different branched avenues that the electrons could go in. They could be shorter, so they could be missing complex 4 or missing complex 3, etc. Or they might have lower PO ratios. Do you remember the PO ratio? So the PO ratio is 2.5 for NADH in a eukaryote and a lot of prokaryotes as well. It is also, or aerobic prokaryotes, I should say. NADH, the PO ratio is 2.5. FADH2, the PO ratio is 1.5. Do you recall why? Every time NADH drops off its electrons, those electrons flow. That ends up pumping out 10 protons. Those 10 protons returning back provides 1 ATP to the cell. Does that make sense? So four protons coming back provides 1 ATP. So 10 protons pumped, 4 are required for ATP. 10 over 4 is 2.5 PO ratio for NADH. With FADH2, don't forget FADH2 drops off its electrons at complex 2. So FADH only powers pumps 3 and 4 to pump 6 protons. It still requires 4 to make ATP, so 6 over 4 is 1.5 PO ratio. Does that make sense? So the PO ratio, again, is the number of ATP that are formed per oxidation of NADH or per oxidation of FADH2. During NADH oxidation, the electrons flow through complex 1, 3, and 4, powering the pumps to pump 10 protons. It takes 4 to make ATP. 10 over 4 is a PO ratio of 2.5. FADH2, on the other hand, drops off its electrons at complex 2, bypassing complex 1, and through electron flow through complex 3 and 4, you only pump 6 protons. It still requires 4 to make ATP, therefore 6 over 4. The PO ratio of FADH2 is 1.5. Maybe you know that for the exam. This is another important thing for the exam. I want you to know what the proton motive force is used for in eukaryotes versus prokaryotes. Remember, in eukaryotes, the proton motive force is used only to make ATP with oxidative phosphorylation in the mitochondria. However, in prokaryotes, the proton motive force is basically protons coming in from the outside of the cell, right? So those protons can be used to drive ATP synthase activity through oxidative phosphorylation. They could be used to drive the motility of the bacterial flagella through rotation of the flagella, and they can be used to drive substances into or out of the cell through active transport. All right. Now, I want you to know about fermentation as well. I want you to know that fermentation involves only glycolysis. I want you to know that fermentation yields only 2 net ATP. And I want you to know that the reason you end up with fermentation products is because you need to replenish something. And I need you to know the thing that you need to replenish in order to keep fermentation from stalling out. I need you to know that thing is NAD+. NAD+ is a limited resource in cells. NAD+ is constantly becoming reduced to NADH during glycolysis. So you need a way of replenishing NAD+. The way you replenish NAD+ is by taking NADH and using it to reduce either pyruvate directly back into NAD+. So you regenerate your NAD+. Pyruvate then becomes reduced to lactate or lactic acid, or by doing the same thing but modifying, reducing a byproduct of pyruvate. So either reducing pyruvate directly or indirectly by reducing a byproduct, a metabolic product of pyruvate. All right. Lastly, phototrophy. What I'd like you to know is about photoautotrophs versus photoheterotrophs. Photoautotrophs, they get CO2 for biosynthesis, whereas photoheterotrophs, they get organic compounds for biosynthesis. Chlorophyll-based phototrophy is an example of photoautotrophs, where you're using light energy with chlorophyll or bacteriophil, depending on if you're a eukaryote or a prokaryote. You then use your you use that for energy and for collecting ATP. You also get your CO2. You get your biosynthesis carbon from CO2. Right? You get your carbon from CO2. During rhodopsin-based phototrophy, on the other hand, you get your carbon from an organic source. Lastly, let's talk about photosynthesis. What do I need you to know about photosynthesis? These are the things I would like you to know about photosynthesis. The electrons are originally obtained from water. You're getting your electrons from water. Water is oxidized to oxygen. That's why plants produce so much oxygen, because water is oxidized to oxygen, and plants release oxygen into the atmosphere. The electrons then go from complex photosystem 2 through an electron transport chain called cytochrome B complex, and then they stall out on complex 1 or photosystem 1. The electrons then go to an enzyme called NADP plus reductase. NADP plus gets reduced to NADPH. Okay, so remember the whole point of this: the electrons are flowing through this electron transport chain. This electron transport chain pumps protons from the stroma, the fluid outside of the granum, into the thylakoid lumen, so inside of these stacks. That proton concentration builds and forces its way through chemiosmosis, exerting a proton motive force, driving ATP production. And this ATP is produced via photophosphorylation. Okay, so just to review, what I need you to know is that during photosynthesis, the light reactions, the products are oxygen, NADPH, and ATP via photophosphorylation. So again, the products are oxygen, NADPH, and ATP via photophosphorylation. That's what I need you to know about photosynthesis. And then Calvin cycle is the other prong of photosynthesis. I want you to know about Calvin cycle, where you're using the ATP that you formed during photophosphorylation. You're also using the NADPH that you formed during the light reactions of photosynthesis in order to conduct Calvin cycle. And so how does it work? I would like you to know these things about the Calvin cycle. First off, that an enzyme, a very important enzyme called ribisco, this is the enzyme. Ribisco attaches carbon dioxide from the air to a carbon acceptor called ribulose bisphosphate, shortened RUBP. Okay, so ribisco is the enzyme that conducts what's known as carbon fixation, taking CO2 out of the air, combining it with RUBP to fix it into 3-phosphoglycerate. Okay. Next, what I need you to know is that's how you captured your C's and O's, your carbon and oxygen. Next, you have to reduce the carbon and oxygen to sugar. So you utilize the NADPH and the energy from ATP from the light reactions of photosynthesis in order to reduce your carbons and oxygens to sugars. First, you form glyceraldehyde 3-phosphate. Then you add 2 of those together to make glucose and other organic compounds. This process is known as reduction. Reduction because you are reducing the carbon dioxide to sugar. You're reducing it. You're taking these hydrogens from NADPH and handing them to the carbons and oxygens to make sugar. Lastly, I need you to know that you must replenish, you must regenerate your carbon acceptor, your CO2 acceptor, ribulose bisphosphate, RUBP. That's the third phase of the Calvin cycle. Again, what do I need you to know? Rubisco, the enzyme, conducts carbon fixation, combining CO2 with RUBP. Next, that's called carbon fixation. Next, reduction is occurring where NADPH is utilized to reduce those carbon dioxide with the help of ATP to form sugars. And lastly, you must regenerate your CO2 acceptor, ribulose bisphosphate, RUBP. Okay, so that is about it. We did it. That's exam 1 review. Chapters 1, 2, and 3 will comprise the material on exam 1. So study hard. You can do it. I know especially chapter 3 can be a little bit difficult, but I'm sure you can do it. Don't forget, again, at the beginning, I do have these videos for nice review on those concepts. You can do it. Study hard. Let me know in the comment box below if you have any questions, and I will catch you guys next time.
Dr. D, Dr. D, Dr. D. Dr. D, Dr. D, Dr. D. A Dr. D, Dr. D, Dr. D. Dr. D, Dr. D, Dr. D. A Dr. D, Dr. D, Dr. D. Dr. D, Dr. D, Dr. D. A Dr. D, Dr. D, Dr. D. Dr. D, Dr. D, Dr. D.